This video is going to present the evolution of the female reproductive system in vertebrates. Obviously, many of the features which we are obviously concerned within the higher vertebrates, mammals, primates, humans, did not first appear since obviously reproduction has occurred long before there were mammals or primates or humans. Sexual reproduction and meiosis goes back to the protists, so eukaryotic cells 2 billion years ago. And protists today reproduce sexually, even though there are some various mechanisms which are unique to ciliates or unique to diatoms here. But one thing that, in addition to meiosis, we can see that the gametes can be flagellated, and that those which aren't, which we would call then the ovum, is non-motile and larger than the sperm. And so this aspect appears early on. Once we start looking at animals, as you can see here in this sponge, the cells which are producing the female gametes, the ova, well, some are here, some are here, some are here. They're distributed throughout the body. So there's no gonad, there's no ovary in that sense. Oogenesis can occur anywhere in the body. That would change in the next group of animals. As you can see in this hydra, this area here is producing ova, but they are segregated in one part. So there is a gonad, and in worms, this then would become an organ. And so as we go through these invertebrate groups, there are some advances in reproduction. Looking at the deuterostomes, which are more closely allied to the vertebrates, there are a couple of features of interest. The main one being that the last meiotic division of the secondary oocyte does not occur until fertilization. So in other words, that meiosis involves two rounds of cell division. So you go through prophase 1, prophase 2, prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. But at ovulation, and this is the case in humans, this is not technically an ovum yet. It is a secondary oocyte. And from then this secondary oocyte, then after fertilization, it then goes through anaphase 2 and telophase 2, divides, and one of the two daughter cells becomes an ovum. So the ovum or egg is not produced until after fertilization. What was released at ovulation is a secondary oocyte, almost done meiosis, but not quite. And this actually was the characteristic of the deuterostomes and chordates prior to when there were vertebrates. Also note that this feature we see here in humans, we also observe in primitive deuterostomes, where there are cells of the ovary called follicular cells, which surround the oocyte, which is undergoing meiosis. And so unlike sperm, which are relatively independent, the oocytes are developing as kind of a group with epithelial cells from the ovary helping them. Now, these early chordates and deuterostomes are quite different. They have segmented gonads. They can be hermaphroditic. And so that's different. One of the things that we will see as we move forward is, unlike the situation in males, where when a sperm is finally released from the seminiferous tubule, when it's made, it's in a lumen, it's in a tube already. So as it matures, it then becomes ready, it develops its flagellar tail, and it leaves into this lumen. And then all it has to do is follow the straight line. It's already in the tube that it simply needs to follow to get to its destination. That is not the case here. So notice where the ovary is. This is the body cavity, the ventral body cavity, and this space is known as the coelom. When the oocytes undergo ovulation, they are not immediately in an oviduct, which will transport them elsewhere. They are released into the coelom, which we'll see in a couple of places, then becomes a challenge because they obviously then have to be taken up from the coelom and delivered properly. And so here you can see a jawless fish. This is a lamprey. Here, and this can occur in other groups as well. The two ovaries actually fuse to make a single one. And here you can see hundreds of thousands of eggs in these female lampreys. Each dot is an egg. They do not pass into an oviduct. There is no reproductive tube which transports these. They're simply released into the body cavity, and then near the cloaca, taken up by a pore, which will then pass them into the cloaca. By the jawed vertebrates, the ovaries are paired. The cells of the oviduct help to take them up in most cases and deliver them through a reproductive tube, and cilia help the movement through the oviduct. So let me just pause and and let me just say one word about the oviduct. So we have different types of ducts which are forming, and so there is what is known as a Mullerian duct, and then a Wolffian duct, which is derived from the mesonephros and its duct. What will happen starting in the jawed vertebrates is that the oviduct, and then and potential modified areas, which could be called, the uterus, the vagina, they develop from the Mullerian duct, which only females have. And that then would be different from the ductus deferens, which develops from the mesonephros here in blue, the Wolffian duct that the males have. Now, obviously, in the evolutionary sense, it's nice to talk about progression and progress. So this gets added, and more or less, and at some point we get to mammals, and then primates, and then ourselves. But then it's obviously more complicated than that often. So, for example, if you were to talk about jawed vertebrates, represented by sharks today, the most primitive jawed vertebrates, a problem is that sharks can differ wildly in how they reproduce. So, for example, here is the ovary, and then here's the opening of the oviduct, and this is the funnel region. There is a shell gland where the shell can be added. There's the isthmus. There can be a region which is called the uterus, although it would be distinct from the uterus in mammals. But if you look at some of the variations in sharks, the ovaries are usually paired, but they don't have to be. They can be single. The size of eggs can vary. Eggs can vary. Some of them aren't meant to be fertilized. In some sharks, many eggs are produced to be nutrition for young shark pups, which help them develop. In some sharks, two funnels may fuse, or one funnel becomes larger than the other. The shell gland can vary in its secretions. Some females can store sperm, something which I will get to. And when we look at the mode of reproduction, it can vary. So all sharks practice internal fertilization. The male pelvic fins have what are known as claspers, which have grooves which help the sperm be delivered inside the body of the female. But some sharks lay eggs where the ovum is released by the ovary. It's taken up here. It then passes through the uterus into the cloaca, and the egg hatches. It's fertilized by the sperm while in the oviduct. So these sharks would lay eggs that then hatch. So this is known as oviparity. But there is another type known as Ovo Viviparity, where the eggs actually hatch while inside the body of the female, and the young are born alive. So the eggs would hatch here, and the young are born. They might receive some sort of nutrition there, and that then gets us into the final type, because in addition to various types of yolk sacs, which sharks can have, so it can be a yolk sac, it could be yolk in a distended stomach. Some shark pups are eating their siblings or eating unfertilized eggs, but there could also be the fetal tissue, which contacts the maternal tissue, which is actually then a placenta. So even though we think of placenta applying to mammals, some sharks develop a placenta, and the young can be born in a more advanced state. And so this obviously then makes it difficult, from an evolutionary sense, to say, this was the progression of female reproduction, because obviously in a group such as sharks, there's such variation. And then the same could be said of amphibians. Amphibians can vary wildly in how they reproduce. In fact, it's my opinion that there's greater variations in reproduction in amphibians than in any other group of vertebrates. Females often produce a large number of eggs at one specific time of the year. So here you can see the ovary outside breeding season and in breeding season, where the eggs are being produced, and there's a huge difference in size. And the same thing is said of a salamander. There's an ovary in a salamander outside breeding season, and here is within breeding season. So each of these dots is an egg, and here is the ovary where they are maturing. In amphibians, the ovaries are small, but get bigger during breeding season. And the eggs are released once again into the body cavity, at which point they have to be taken up by the oviduct in order to be transported. If they are not taken up by the oviduct, then they will be broken down and reabsorbed. And so the eggs can then be stored and in a large distal region known as the ovisac, because very often, male and female frogs will mate, and then at that point, when the male is releasing his sperm, the females release her eggs all at once. But this can vary. So here you can see a salamander. Here's the ovary with the eggs, and then here is the oviduct. Once again, the eggs leave into the body cavity and then must be taken up by this oviduct. Here's the oviduct in the female frog. There's the ovary there. So the eggs then have to be taken up by the opening here. So here you see a typical form of amphibian reproduction where males and females are mating. Fertilization is external, occurring outside the body of the female. So the male will release sperm, the female will release the eggs, and the fertilization occurs there, and then the young obviously are developing in the pond. There are variations. So most salamanders practice internal fertilization, although male salamanders do not have a penis. That only exists in amniotes. What would happen is that a male salamander would deposit packets of sperm that then the females could take up using cloacal lips, and so that the sperm would fertilize the eggs inside the body of the female. In one salamander, the spotted salamander, the female's body can actually have green algae living inside her cells, endosymbiosis, cells of the algae living inside the female salamander cells. And these can get passed into the egg, so that in the egg mass, the algae can be producing energy through photosynthesis and oxygen, which will benefit the embryo. Reproduction in amphibians varies wildly, though, because it can occur in water, giving rise to aquatic larval stages, which breathe through gills, but it does not have to. Many salamander eggs are laid simply in damp, places and undergo direct development. Some frogs will have the tadpoles develop, in specialized pouches on a parent's back or inside the stomach of a parent, which stops eating during this season. And so there is great variation in the reproductive styles of amphibians. One frog actually practices internal fertilization by inverting part of the cloaca. When the first reptiles evolved, the males developed the intromittent organ, the penis, which would allow for internal fertilization. And then its homologue then also appears in females, the clitoris. So the clitoris is a feature of amniotes. Here are water snakes, which are obviously comfortable in the water, but they've come out on to land to mate, and they then practice internal fertilization. And so obviously a significant change in reproduction in the first amniotes. The amniotes then can lay their eggs on land, which means that they are not as dependent on water for reproduction as is the typical amphibian. So here you can see a turtle doing this. And all amniotes, reptiles, birds, and mammals, then have these 4 extra embryonic membranes. The yolk sac, and the yolk sac actually goes back to fish as the energy for fetal development, so that wasn't a new one. But these other 3 are. First, in blue, the amnion, and the amnion surrounds the embryo in fluid, right? So, not only does this apply to reptiles and birds, but also mammals. We developed in amniotic fluid, and even though we are air breathers, after birth, there was this amniotic fluid in our lungs for the first 9 months of our life. Another bag here in green, the allantois can collect urinary wastes. And then the entire fetus is surrounded by this chorion in red. The chorion can perform gas exchange with the outside world. If this is a lizard in an egg, in mammals, the chorion will form a part of the placenta, as I will get to. So you'll notice that here you have an egg-laying lizard that has these 4 extra embryonic membranes, including the amnion, and thus reptiles, birds, and mammals are amniotes, capable of reproduction on land thanks to these extra embryonic membranes. But we'll see the same extra embryonic membranes in mammals, but we will get to that. Reproduction then is follows that standard model in reptiles. Lizards and snakes possess hemipenes, a pair of penises, although only one is used at a time. Some snakes will only develop 1 ovary. And while many snakes lay eggs, many give live birth. Live birth is actually quite common in snakes and actually quite common in lizards as well. And it's not as if, here it evolved once and all of the descendants from this lineage have a live birth and the other ones are egg laying. Like in lizards, for example, you have a family like skinks, and these branches can lay eggs, these other ones can give live birth, and so live birth has evolved multiple times. So some sharks give live birth, some lizards and snakes give live birth. And so, obviously in mammals, live birth is a big deal for the Therian mammals, but it has evolved multiple times. Now, female reproductive tracts can store sperm. And so this is, something which, appears in many groups. And so females may have mated, but may not yet be pregnant, but weeks later, months later, years later might be pregnant. So you might have, a turtle or a snake, isolated by itself for quite some time, and that all of a sudden becomes pregnant and lays eggs because they had mated and had stored sperm. Now that's foreign to us because obviously, in humans, sperm only last a very short time in the female reproductive tract. But as long as they are nourished, if energy supplies are given them, the cells will survive. And so a sperm can be kept. Now, actually, there doesn't have to be a special reproductive structure for this. I'll get back to that. And so, for example, if for example, in dogs, is there a special structure which will hold sperm and store them? No, but the sperm can be associated with the uterine glands of the uterine lining and get nourishment there. There can, however, be specialized structures, these close-ended tubes. So there's one opening, then it ends blindly, which can store some sperm, which are known as Spermatique. These are known in vertebrates like earthworms, mollusks, bees, nematodes, and crabs. In the vertebrates, we see them in some reptiles and birds, but the preferred term rather than Spermatique is sperm storage tubules at that point. Delayed fertilization, the storage of sperm, is also known in the one and only frog, which practices internal fertilization. And so it may be that internal fertilization, is, a bit of a prerequisite for the storage of sperm. And if you were to ask for duration, it can certainly last varying amounts of time, from weeks to months to even years, with the record being 7 years in snakes. And so, sperm can be stored. Going through the vertebrate groups, just to say a few words about birds then. Birds typically have two ovaries, although most birds only have the left one. Raptors still have two, but after embryological development, one will deteriorate. During breeding, it gets much bigger. You'll see the chicken ovary in just a minute, and it will typically release one egg at a time, but then as that egg then moves along the oviduct, gets to places where shells are being added, then another egg can undergo ovulation. So the eggs, which you see here in the ovary, are released one at a time, and they are released into the body cavity. And so here's the ovary, and it has eggs at varying stages of development. The eggs undergo ovulation into the body cavity and are taken up and passed onward. So here's the infundibulum region of the oviduct, where they then get passed onwards. Now, not all eggs are taken up, in which case they should then be broken down by white blood cells like macrophages here. And so this can be okay, although in birds and other reptiles as well, that this could actually be a cause of a physiological problem, and then may even result in the death of the female because of this egg. If it causes inflammation, if bacteria were to infect the yolk, this could actually then lead to serious problems and death. And so once again, there's a problem with the egg not already being in the oviduct the way that we saw with sperm, that it has to be taken up from the body cavity. And so in most birds, only the left oviduct is present. And there are varying regions of the oviduct. And so what happens is after ovulation, The egg will hopefully be taken up by the infundibulum. It may spend an hour here. If mating has occurred, then within 15 minutes of entering the infundibulum, fertilization could occur. The egg then passes into a region of the oviduct known as the magnum, where there are some things added around the embryo. More yolk is added. There are a couple of hours may pass. And then there is an isthmus where the inner and outer shell membrane is added, 1 or 2 hours here. And then a uterus or shell gland where the hard outer shell is added, and the egg will spend about a day here before passing out the vagina and into the cloaca. And so here was just some of the variations in vertebrate reproductive systems. But now I'd like to focus on mammals and obviously move to humans. So the earliest mammals shared features. In addition to hair, they also share mammary glands, which I'll be talking about at the end. So this is a mammal feature, although a number of animals do produce some nutrition for their young. So doves, for example, can produce a crop milk to help nourish their young. But there are specialized sweat glands known as mammary glands, which all mammals develop. And embryonic mammals then develop a mammary ridge, all right? And then different species can develop breast tissue and nipples at along different points of this mammary ridge, depending on what species they are in. The corpus luteum is typical of mammals, which will release the hormone progesterone, which causes cell division and proliferation in the endometrium. So mammals, share a number of features of their reproductive systems. After the lineages of egg-laying mammals diversified, then there was a group of mammals called the Therian mammals. The Therian mammals are those that give live birth. That would include not only the placental mammals, but also marsupials. Also, one of the features, in addition to live birth, is that there would be the sicular development and shedding of the endometrium, so a menstrual cycle. And so if we were to study how this works, there would be some shared features of the embryo as far as the circulation being different, so that it can now be taking up oxygenated oxygen from the placenta rather than from the lungs. So there are circulatory changes that both marsupials and placental mammals have. Also, some immune adaptations, although the marsupials give birth at a very young state, and then the young have to complete development very often in a pouch. So here you see an opossum's pouch, and notice that there are nipples in the pouch, where the embryos, which are very young when they are born. So a kangaroo's infants may only be the size of, say, a bee when they are born, and then complete their development in a pouch. The Therian mammals, or the placental mammals, they then modify their immune system so that the developing infant would be tolerated inside the body of the female for a longer period of time. And so once again, here in the opossum, the endometrial lining of the uterus gets shed periodically. Okay. Now, many of the external structures of the female reproductive system develop later. So the labia majora and minora are known in primates, and the most primitive mammals still possess a cloaca, where there is a shared opening for the urinary, reproductive, and digestive systems. And so, we are accustomed to, the urethra, the vagina, and the anus being, completely separate, but they can empty into a shared region known as the cloaca in more primitive mammals, or say the urethra can empty into the vagina. So if we then look at the group of mammals, which we call the placental mammals or the Therian mammals, the fetuses are maintained inside the body of the female and much longer, so they can reach an advanced state. So here you can see the uterus of a cat, which is not pregnant, and then the uterus of a cat, which is pregnant. And you can see, the marked increase in size and the structure because the young will be born at a very advanced state, so not pregnant in this cat. So this is the uterus, once again, in the cat, and that's a kitten. That's a kitten. The young are obviously, being born in an advanced state. And we see that, typically, here's the uterus of a not pregnant cow and a pregnant cow. The embryos developing inside then the uterus, wrapped in those extra embryonic membranes. Here you can see a pig which is not pregnant, and here you can see the uterus of a pig which was pregnant. Now, in the most primitive mammals, the uterine tubes are two separate tubes which do not fuse at any point. Whereas in most mammals, there are ovaries, there are oviducts, and much of the uterus forms these two separate uterine horns, but the uterus does then fuse and form a body which is then shared. And so the uterus of most mammals is Y-shaped, where there are these two uterine horns where the fetuses can develop. So we see that here in the cat, and then here in the cow, there's the body of the uterus, which is fused, but here you can see the space in the two separate uterine horns. Here you can see in the pig, the ovary, the oviduct, and here's one of the two uterine horns. Once we get to primates, now the uterus then is different, and a more of a pear-shaped uterus forms. So once again, if you were to look at a cow or a cat, you would see a Y-shaped uterus, whereas in humans, it actually begins as a Y-shaped uterus in embryos, but then changes to become more of a pear-shaped uterus, as we can see in this monkey. So here's an ovary, here is the oviduct, here is the pear-shaped uterus here. Some women have the abnormality of having a Y or a T-shaped uterus because that modification doesn't occur during embryonic development. Once again, here's the ovary, oviduct, and uterus in a monkey. Now, in primates, the urethra empties into a space known as a vestibule, separate from the vagina. And so there is a complete separation in female primates, and that would include humans, of the urinary and reproductive organs. But in many mammals, that's not the case, where the urethra can, say, fuse to the vagina at its distal portions. And so the urinary and reproductive systems do not have to be 100% separate as they are in mammals. I'm sorry, as they are in primates. As in the group of apes, there are labia majora and a menstrual cycle of about 30 days. And so clearly then, there have been, gradual changes in reproductive systems over time. Before focusing on humans, just a couple of final things. I've got videos for students who are trying to identify structures in the cat, which are going through the various structures in the cat. Then once again in the pig and the cow. One can see, just once again, the advanced state at which placental mammals give birth to their young. So here's the ovary, the oviduct, the oocytes are released into the body cavity. They must be caught by that infundibulum and transported into the oviduct before going into the uterine horn. Right. And so here, the female reproductive system is anchored by ligaments. So there is an ovarian ligament anchoring the ovary. There is a round ligament and a broad ligament, which will attach to the uterus. Here you can see the uterus in a pig. You can see the difference in the pregnant versus not pregnant. And then inside the uterine horn, the fetus would be developing in its extra embryonic membrane. So here you can see multiple fetuses forming there. Here's one wrapped in the extra embryonic membrane. Now you'll see this in this next video and also in the following one. There are different types of placenta which can form. And so if you were to look then at the cow, the maternal tissue does not completely surround the developing fetus. Instead, there are very specific regions known as caruncles where you have maternal tissue and then fetal tissue which grows into it. And so unlike what we'll see presently, in humans. Here, the entire fetus is not surrounded by a placenta. The placenta is only the connection between these these fetal cotyledons and the caruncle made in the uterus. So that's something that we can see in some of the hoofed animals here. So once again, even though when we speak of the placenta, and placental mammals, a lot of the modification has then changed. So here you see the fetal calf, and then its cells would be touching the mother cells. Here are these caruncles where the nutrition would be relayed. So once again, we speak of placental mammals, although it should be stated, there are different types of placenta. And as I had mentioned with sharks, you can actually have different types of tissue being a place where fetal cells are touching maternal cells. So technically, that's the definition of a placenta. If you just have fetal cells touching maternal cells, and then materials can be exchanged, which help the fetus during its development. There can be placentas which are known in some fish, some amphibians, etc. The type of placenta which the eutherians possess is what's called a chorioallantoic placenta, in that the chorion, that's the extra embryonic membrane in red, these are the fetal cells which are touching the endometrium of the uterus. But blood vessels from the allantois, here in green, they then also contribute to the placenta. And so the placental mammals, that is what we mean when we use the term placenta. Right, so I'll go to this next one. And let me just show you this under a microscope. This will vary, but let's talk about what we would see in primates, or most primates at least, I'm sorry, and humans, where there are the cells of the endometrium of the female, and they can form glands and blood vessels. Then bleed and ooze blood onto the chorion. So this maternal blood does not enter the fetus, and maternal blood and fetal blood do not mix. The mother is simply going to ooze blood onto the surface of the chorion, and the chorion has these projections known as chorionic villi. And thus, if the maternal blood is oozing over the chorionic villi, here you can have transport of substances. So oxygen from the maternal blood can then diffuse. So here's the maternal blood in a space known as an intervillous space, and here is the fetal part of the placenta, the chorion. So oxygen can be going from the mother's blood to the fetus. Carbon dioxide can go from the fetus then to the mother's blood to be lost. Glucose, amino acids, ions can diffuse to the fetus from the mother's last meal. Urea produced in the fetus can diffuse so that the mother's urinary system may release it. And then obviously we're concerned about a mother's diet because things like drugs, alcohol, viruses can also pass through the placenta. Let me just once again go down to if one were to study diverse mammals, there are different types of placenta. And so the marsupials, they give live birth. But we do not call them, quote, placental mammals. And in a sense, that's wrong because they do form a placenta. They just form a different type of placenta where it is the chorion and blood vessels from the yolk sac, not the allantois, which then form the placenta. So marsupials, even though we don't call them placental mammals, they do form placentas, just for the most part, a different type of placenta, which uses blood vessels from the yolk sac. There are a few marsupials who actually do form the same type of placenta as we see in so there's the allantois. Its blood vessels can contribute to the placenta. So most marsupials form a choriovidulan placenta, where it's blood vessels from the yolk sac. And while the, quote, placental mammals, it's blood vessels from the allantois, there are actually a couple of marsupials which actually form a chorioallantoic placenta like the placental mammals. As I had mentioned, if we're just defining the placenta as places where fetal cells and maternal cells can touch, look at all of the different structures which can help to form this placenta. So it can be, uterine villi, it can be gill filaments, it could be the pericardium of the infant, it could be, just a whole host of various things. And so, biologically speaking, I often use the term placental mammal, but once again, marsupials, they have a placenta, just typically a different kind. And there are then salamanders and fish and frogs and sharks, which also then have a form of placenta. And so there's a lot of variation here. One of the other forms of variation is obviously the maternal strategy. And the maternal strategy then can simply then ask the question, what is the best way to have the maximum number of genes spread to the next generation. And one can see some females are producing sometimes hundreds of thousands of eggs at a time, but no care will be given to these individual ones. And so that's one strategy. Here you can see in the frogs, obviously lots of eggs are laid at once. Whereas if one looks at, birds, you can have, a great deal of yolk given. So a much smaller number of offspring, but now each one is going to have a much better chance at survival because of the increased nutrition given them. And then obviously in mammals or Therian mammals and placental mammals, this is even more the case where the young will develop for a long period of time inside the body of the female and receive substantial nutrition. So now with that, I could focus specifically on some of the features of reproduction in humans. But given the length of this lecture and the audience, students studying comparative vertebrate anatomy, I'm not going to go into some of the cellular aspects the same way I would in, say, a human anatomy and physiology class. So I'll just point you out to a number of videos. I'm just going to give more of an overview of this next part. So in meiosis, which is occurring in the ovaries, that's the main job of gonads to produce haploid cells. You would think, when we would begin teaching it like this, that one cell would undergo 2 rounds of cell division and produce 4 haploid cells. All right, well, that is not the case. We see a number of differences in meiosis in females compared to males. One is the size. Look how big the oocyte will be compared to the sperm. Look at the follicular cells. The oocyte will begin as part of a team, as opposed to the sperm developing singly. But now also, as you'll see in a second, when the primary oocyte divided, it made 2 daughter cells. This is one of the 2, and this is the other one. You'll see that the cell divisions were not equal, producing a secondary oocyte and a polar body. At fertilization, then the secondary oocyte divides, but once again, this division is not equal. And so another polar body is produced. This first one may or may not divide. And so now you produce 1 gamete. So this is the egg. Meiosis in males produces 4 sex cells for every cell that begins meiosis. Meiosis in females produces 1 sex cell, 1 egg, and then 2 or 3 of these smaller polar bodies, which will degenerate. And so that is a major difference. And so instead of making 4 haploid cells, which can function in reproduction, it is only the first. So let's just watch that again. In oogenesis, which is the making of an ovum, the cells undergo prophase 1, metaphase 1, anaphase 1, telophase 1. Once again, it is in prophase 1 that 2 very important genetic processes occur: crossing over of genetic material and the independent assortment of chromosomes. Note that both of the pink chromosomes don't have to line up on the same side of the metaphase plate. But then, as I said, this division is not equal. The majority of the primary oocyte's nutrients ended up in this secondary oocyte, not in the polar body. And then the second round of cell division occurs again, and then there will be 1 ovum that receives all of the nutrients, and then these other polar bodies which will degenerate. Why? Well, in humans, for example, the ovum and its cytoplasm, these are the only nutrients that an embryo will have during the first week of its life. And so therefore it's good then that all of the resources end up in 1 cell to produce 1 high quality gamete rather than multiple ones. And I'm sorry, it just froze. All right, I apologize. I'm waiting for this to catch up. I'll just keep going. So the ovum. The ovum then has the nutrients which are going to carry the embryo through the first week of of its life. So I apologize, I keep talking. I'm hoping this is still recording well. In the okay, I apologize if this is not working out well again. So let me just sum up by saying that there's far more than to this, and I have a separate video where it will go through the stages of development of the oocytes in women, which actually begins before birth and isn't done until fertilization. So it takes an incredible amount of time. And then also I have a playlist on milk and the mammary glands, which then release them. So. Okay, so I might be back. So, so I apologize if there were problems in that. And so I have a playlist, and certainly there are so many steps in this. So, for example, the germ cells for the ovary, they don't actually form in the ovary. They have to travel there from their original place of development in the yolk sac. There are stem cells known as oogonia, just as there are spermatogonia in males. But one of the big differences is that the oogonia begin meiosis before a woman is born. Let me repeat that. While spermatogenesis, this occurs after puberty, oogenesis actually begins before birth, and then a woman's number of oogonia then is then set at a final term. So from the 4th to 5th month, there are 6 to 7 million oogonia, which reduce to 1 million primary oocytes, which actually have begun meiosis by the time of birth. So women actually start to make gametes before they are born, but then they just pause. And so that's another difference between the process of meiosis in males and females is the timing. In men, it begins and ends in a short period of time. In women, it actually begins prior to birth and then pauses. So one of the early stages of prophase 1 is initiated, and then there are these primordial follicles where these oocytes are then surrounded by these follicular cells. And so there are these primordial follicles which will stay in the background during a woman's reproductive life. Then at some point following puberty, then a primary follicle will then become a secondary follicle. Now, in the lamprey that we saw, obviously the female was laying lots of eggs all at one time, whereas in humans, obviously that is not our life cycle. And so these will occur associated with the menstrual cycles at various points throughout a woman's life. We can split these follicles into various stages. I go through these in videos. I'll just summarize it. Here, the number of follicular cells can then increase as it becomes wider. And at some point, a space called an antrum develops in the follicle, which will be filled with a follicular fluid. And this follicle then is not only getting larger, but it's now called an antral follicle. There are so many signals going back and forth. Most of these are very local signals where one cell layer is affecting its neighbors. rather than these being hormones which are traveling a great length. But there are then hormones which are coming from the pituitary, which are important as well. So FSH coming from the pituitary is the follicle stimulating hormone, which will then stimulate some of the last development of these follicles, which could actually be migrating through these phases for about 2 years before the month where they will undergo ovulation. This follicle can be rather complex. Once again, here's an antral follicle with the antrum, and you can see all of the layers here. And while the follicle will produce estrogen, it actually then seems to be here in orange, the theca interna cells, which responds to the pituitary hormone LH, will make cholesterol into androgens, which are then taken up by these blue cells, which respond to FSH to make the androgens into estrogen. All right, so it's actually complex. At the midpoint of a menstrual cycle, the pituitary spikes in its production of what's known as LH, or luteinizing hormone, and its luteinizing hormones spike, which then causes ovulation, the release of the secondary oocyte from the ovary. And so the development is actually quite complex, and I go through it individually in these videos, but LH will cause the smooth muscle cells, which are outside the follicle, to contract. This increases pressure, and this will cause this then to burst, releasing the secondary oocyte into hopefully the oviduct. So here you see ovulation. Luteinizing hormone causes this to constrict, and this is released into the body cavity. It's not automatically in the oviduct, but the oviduct has an expanded opening, the infandibulum, which hopefully will then take this up. These cells which remain, they form an endocrine structure known as the corpus luteum, or yellow body, and this then secretes the progesterone, which will help to maintain the uterine lining in case of pregnancy. So here you see a corpus luteum, and under the influence of its progesterone, the endometrium gets to be its thickest. Now, like I said, the oocyte does not have to be taken up by the oviduct, but if it was not, then an ectopic pregnancy would be possible, and this could actually be life-threatening for the woman. And so the infandibulum, the end of the oviduct, has these finger-like projections known as fimbriae, which help to take up the oocyte. The cells of the oviduct have cilia, which help to move this secondary oocyte towards the uterus, and then there are also muscular contractions in the wall of the oviduct as well. So these three mechanisms are moving the oocyte towards the uterus. The ovary certainly undergoes changes during a menstrual cycle, but so too does the uterus, and we can refer to the uterine cycle. Much of the uterine cycle will involve the innermost lining of the uterus. The uterus has an outer perimetrium, a myometrium where the smooth muscle is, and then an endometrium. This endometrium has a basal layer here in gray, which is always present, and then under the influence of estrogen after menstruation and its menstrual phase, in which the endometrial lining of the previous month is shed. Now a new endometrial lining can begin to develop. So here you see during the menstrual phase, the previous endometrium is being shed, and then under the influence of estrogen, so this vesicular follicle developing then makes estrogen, which causes the endometrial lining to proliferate. This causes the proliferative phase of the functional layer of the endometrium. And then after ovulation, the corpus luteum releases that progesterone, which then causes not only the endometrial lining to reach its thickest point, but also causes the endometrial glands to start to release nutrients. So this is known as the secretory phase because of the secretion of these uterine glands. Then finally, I have some videos on mammary glands where I go through their formation, and then just how complex milk is. Milk has proteins, milk has lipids, milk has carbohydrates, milk has hormones known as lactokines. There are microRNAs where the mother's body is actually sending commands to the infant and changing the expression of genes. There can be bacteria from the mother's skin, which helps to establish a healthy milk or a microbiome in the infant coming through milk. So I have videos which go through that if you are interested. And then also fetal circulation is different from adult circulation. And I have a number of videos on that as well. And so this has then been a summary of the reproductive system in female vertebrates. I have separate playlists and videos which are more human specific if they are of interest.